My name is Cristobal Uauy. I'm a wheat geneticist and a group leader at the John Innes Center. I started this research area in terms of wheat genetics and genomics because I've always been fascinated in producing enough food to feed a healthy planet. I think that with these technologies, we can actually affect food security. This research is very important because across the world, everyone eats wheat or pasta or noodles or naan bread. So all civilizations, all societies eat wheat. And they eat a lot of wheat. Every day, each one of us will eat 50 plants of wheat. So we need to produce a huge amount of food. And not just produce food, but also has to be healthy and nutritious. So these are kind of the challenges we face. And of course, we need to do it in an ever-changing environment with climate change, less use of pesticides and fertilizers. So it is a challenge that we need to not just improve or increase our production of healthier food, but also decrease its environmental footprint. So that's one of the challenges we face. The work that we're doing is trying to combine genetics and genomics with the biology of the plant. And we're looking at yield. And that means that we're trying to understand what are the genes that make the grain heavier or longer and wider, because this influences how much yield we produce in the field. And yield is very complicated, just like intelligence in humans, many genes affect yield. So we're trying to break it down into these smaller components. And what we found is that we can find genes that affect the width or the length of the grain, and by using natural variation, that means variation that's already out there in the fields. And we find that we have differences of about 5%, which is important, but it's not going to get us to that challenge of 30 or 40% increase that we need to achieve. So in order to do that, what we've learned is that when you look at other species like maize and rice, these species with one gene, you can get perhaps 20 or 30% differences in yield. And in wheat, you can't do that because wheat is a polyploid, meaning that it has 3 copies of most of its genes, unlike rice or maize that have only 1 copy because they're diploids. So in wheat, you have 3 copies. And what we found is that many of the genes that regulate these yield components are actually negative regulators. That means that they put a molecular break. They stop the grain from growing. So in wheat, when you take 1 break off, you still have 2 breaks. And that means that your effects are quite subtle, 5%. But interestingly, what we found now is by being able to generate variations in the 3 breaks at the same time and combine them in one plant, all of a sudden now we have effects of 20 to 30% increase in the grain size, similar to diploids. So this is variation that no one has ever seen before. It doesn't happen just by chance that a plant will have these variations together, but by using genomics and DNA, we can actually find the mutations, combine them, and we have plants that have much bigger grains, and now we're testing if they will have higher yields in the field. The field trials we're conducting at the moment is trying to understand this genetics that we've done in the glass cells and see how it works in the field. And that's a critical test to do because a plant can perform very well in a pot, but that's not how it grows. It grows in farmers' fields, so we need to put it in the field to see how that behaves. So how will this increase in grain size actually behave when it's across a whole hectare? Will that translate into yield? Will that yield actually have lower quality, or can we actually maintain the protein and micronutrient content of the grain? So testing the plants in the field is the ultimate test before that goes to the breeders to make new varieties. The research we do is generally long-term, means that it might take 5 years to do a discovery or even 10 years to do the discovery, and then that needs to go into a variety that goes to the field. And to make a new variety, it might take 8 to 10 years, and we work with breeders to do that part of the process. So it might be 15 to 20 years before our discoveries get to the field. It's fascinating right now what we can do in genetics given the new genomic technologies. We've seen the revolution in humans, and now that revolution is coming to plants. And as we look forward, plants will become more and more important in people's lives because we realize that actually instead of trying to cure people with medicine at the end, perhaps we can give plants or we can give food as our medicine in the future. And I think there's that realization. So being able to have young scientists making these improvements in plants will really not just help the environment and food security, but also help the health of people around the world.